Hello, good evening, and welcome to the South Dakota Discovery Center STEM Savvy Teen Science Cafe. And my name is Bree Oatman, and I'm the Education Director at the Discovery Center. And tonight we are joined by Dr. Adam Hoppe from South Dakota State University. And we also have two of our Teen Science Cafe youth leaders with us, Mesa and Race. And hopefully we have a bunch of people signing in on YouTube and on Facebook and on Twitch. So if you're watching us, feel free to set up a watch party and spread the word to get more people involved. But we, every time we have one of these cafes, basically the leadership team comes up with a topic of something they're interested in learning about, and we invite an expert to come in and share their career with us and to talk about what they're doing. And usually we do this in person, and there's usually food and hands-on activities. And so we are kind of experimenting with how to do this virtually. So tonight we're going to have a presentation, but we're also going to be playing a video game and trying to get people to play that together. There's a Kahoot, and at the end there'll be a survey. So if you're joining us from Facebook, YouTube, or Twitch, please feel free to ask questions via the chat or to comment, and we will try to field those questions as they come up. And with that, I'm going to hand this over to Adam to go ahead and do his start his presentation.
Okay, thank you so much, Bree and Race and Mesa for having me and all the folks at the Discovery Center. I think we're going to have a lot of fun. We can talk a little bit about CRISPR and some of the ways that we're using CRISPR as a technology to better understand the coronavirus that's causing so many headaches for all of us. So I think to start out, I'll talk just a little bit about myself and sort of my journey in science. So I am a professor of chemistry and biochemistry at South Dakota State University. And I like to think of myself as kind of a scientist that does all sorts of stuff, kind of a jack of all trades in science. A person who really uses a lot of interdisciplinary tools and thinking. So I kind of use this map here to sort of show you the course I've charted in becoming a scientist. So I was born and raised in Crookston, Minnesota, up here in the northern part of Minnesota, where this first circle is up here. And I attended high school there and did science and played a lot of sports. And then when it came time to go to college, I just traveled down the road a little bit to Concordia College in Moorhead, Minnesota. And I wasn't really sure what I wanted to do. I was kind of stuck somewhere between music and physics. And I learned pretty quickly it was hard to do both of those things in college. I still sang in the choir and I played the guitar in a band, but I wasn't really able to be a music major and a physics major. So I had to make a choice. And I chose physics because I really love science. I loved experimenting with things. And I studied physics, I studied math, and I studied a little bit of chemistry while I was at Concordia College. And I earned my BA in physics and math. And around the end of my time in college, I met a gal who later became my wife. And she was a biology major. And she would tell me a lot about all these biology things she was studying. And I didn't know really any biology, but it sounded really interesting. And so at that time, I was trying to figure out what to do next with my life. And I applied to a program called Medical Physics at the University of Wisconsin in Madison, Wisconsin. And I traveled there and started trying to figure out how to apply physics to medicine. And ended up taking a lot of classes with doctors. And in doing so, I had to learn a lot of biology. And I really fell in love with biology. I earned my master's in science in medical physics there, mostly doing radiology work, so imaging using PET scanners and CT scanners to try and see inside patients and figure out what was wrong with them, and also try and work on the technology and understand how could we improve that kind of technology. But I was really fascinated with like cells and the insides of cells and molecules and how they all work together. And from there, I was really attracted to a field called biophysics, or molecular biophysics. And I did some shopping around, and I found a PhD program in biophysics. Found a few of them, and finally settled on one at the University of Michigan in Ann Arbor, Michigan. So moved to Ann Arbor and worked on my PhD here. I also stayed on as a postdoc and eventually a staff scientist in the medical school, where I learned more about microbiology and immunology. So, basically, from here to here, I became a physicist, mathematician, sort of an applied physics person, and then ended up really getting interested in more medicine and immunology and cell biology during my time in Ann Arbor. I also had the opportunity, to travel quite a bit around the world and work with other scientists. And probably the most impactful one was I spent 2 stints at the Pasteur Institute in Paris, France. So Louis Pasteur, you may have studied him. The Pasteur Institute was built around his house, his home in Paris, France, where he worked. And I was able to study there for a couple of 3-month stints doing research with some of the best microbiology and immunology faculty in the world. That was sort of back and forth between living in Ann Arbor. And then eventually it was my wife's turn to pick where we went next. And she was also studying in Ann Arbor and earned her PhD in epidemiology, or master's in epidemiology and a PhD in toxicology. And her choice was she wanted to go back home. And home for her was Brookings, South Dakota. And at that time, South Dakota State University had a job opening for a chemist, a biochemist. And I was pretty much a really good fit for that job. So we moved here, and we've been in Brookings since the fall of 2008. It's about 12 years now. In Brookings, I'm a professor. I also run a research lab where I train graduate students. I mentor postdocs, and we do research funded largely by the federal government, as well as a number of collaborations with companies and private industry to try and help develop technologies and therapeutics for some of these companies. So yeah, that's a little bit about my trajectory and my history becoming a scientist. I can talk a little bit about the philosophy and the way I think about science. That spurs any questions. Starting in physics, I was really influenced by this guy, Richard Feynman. He was a real famous physicist. And he was famous in part for his ability to solve physics problems, but also because he had a really straightforward way of thinking about stuff. And this is one thing he said about biology, is one way to solve biological problems is you just look at the thing. And his idea was, if we can build good enough microscopes to see down into the inner workings of biological systems and just see how they work, kind of like this clock here, we can understand them. So we can understand how the gears of this clock fit together, how they move, and if we take it apart carefully, we could probably really get at the inner workings and truly understand this clock. So that's been very much my thinking, or the mindset that I've had as a scientist. And I kind of use this diagram here. So it's kind of the opposite of engineering. So maybe an engineer might draw up blueprints, use those blueprints to build parts, and then put the parts together to build something like a jet airliner. In bioscience, we have to go the other way, right? We have to look at a person, a biological system, a patient, right? And work backwards and understand how things work inside of that individual at the cellular, molecular level, and really dig down deep into the genetics of that underpinning, the blueprints for our lives, which are written in our DNA. So I really look at what I do as having been trained to think this way, but then apply my science going the other way. Okay. And I'll give you just one example of that. So this is an example of a collaboration working with scientists at SDSU and as well at the School of Mines and Technology, where we're creating very advanced microscopes to be able to see cellular processes and molecular processes in 3D and in real time. So what I'm going to show you here is a movie. The gray mesh here is an immune cell. And this immune cell happens to do its job by destroying infected or cancerous cells by eating them. It's like Pac-Man or the Blob, right? And what you're going to see here, you can just see the edge of it in purple, is a cell that is being recognized by this immune cell and eaten and destroyed. So here comes this movie. You see the immune cell recognizes this target cell and it extends its body around it and wraps it up and pulls it into the inside of it, where it will digest it, much like if you ate a cheeseburger. Okay, so my lab is really interested in trying to understand how this process can be harnessed and used therapeutically to destroy cancer cells, cells that are infected with viruses, which will be much of the topic today, and cells that are just not functioning properly in the body. So I'm going to use a little cartoon here to kind of illustrate some of the ways we think about the big picture of the science we do. I'm pulling some of these images from Cells at Work. That was a Japanese anime, and I guess now there's a Netflix show that illustrates some of these. But moving our conversation now into how viruses work and what they do. So if you think about the cells that make up the body, you've got cells that are all doing their jobs, like this guy, and homeostasis, so how they're functioning most days. Some of those cells can become infected with a virus, turning them into most of those infections in human cells. They become zombies, or they will produce viruses, replicate the virus. I'll talk about that in a second. But it really changes their function. So instead of doing their normal job, these cells are now walking, mindlessly through the body or doing mindless things in the body. And the body can quickly recognize that and mount an antiviral response. And I've written interferon here. That's one of the signals that your body will use to tell the cells we have a problem with viruses. And it's also what makes you feel fairly crummy when you have a viral infection. It can give you a fever, stuffy nose, sore throat. Those are all associated with this interferon response. But like this picture, it puts a damper on these viruses. It doesn't destroy them though. So usually it doesn't destroy them fully. So for that, we need other immune cells, which when life is good, they're just hanging out and helping the body along. But when they sense these viruses, they become activated. And in their activated state, they can be really potent zombie killers. And so an ideal immune response, like to COVID, you get some cells that get infected, they get recognized. The antiviral response, like put some cold water on them, slows them down. That gives enough time for the immune cells to become activated, and for other immune responses, including things like antibody responses, to kick in, and then they can start attacking these infected cells and removing them from the body. And in some cases, sometimes the immune cells can also become infected with viruses, in which case they can be fairly damaging, like this example of Harley Quinn here. So really, this is the kind of questions that my lab, we don't always use cartoons, but these are the kind of questions that we try to address and understand how viruses function inside cells. So I'm going to tell you about a little bit about CRISPR and about how there's a relationship between CRISPR and viruses naturally. And I'll talk a little bit about that, and then I think we're going to play a video game to try and get a handle on, have some fun and get a handle on CRISPR. So pretty much all viruses have to infect a host cell. So they are not alive. We don't really think of viruses as alive. So they're sort of like rogue pieces of information, okay? And they have to infect a host cell. Generally, they're fairly specific. So if it's a human virus, it usually will only infect a human. A cat virus usually only infects a cat, cow virus, and so on and so forth. Every once in a while, there are some viruses, but very few, that can go between animals. There aren't very many. And every once in a while, a virus can jump from one species to another, which is partly what we think of, part of what we think has happened with the coronavirus and the COVID-19 situation. So some viruses, we'll see in a second, we'll talk about a bacteriophage. They can infect a cell, and when they do, they will replicate in that cell until they kill it. Usually they explode, release the viruses. Those are called lytic viruses. Some viruses don't kill the host cells. Instead, they're more like that zombie example I was just showing you. They'll infect, they replicate, and they kind of take over the host cell and use the host cell to assemble new viruses and keep shedding those viruses, which can then go on and infect new cells. And then there are also some really interesting viruses called the retroviruses, which they can infect a cell, and then they can also insert their genome into the genome of the target cell permanently. So they can basically write a copy of their blueprints into your blueprints, so the cells that they've infected. They come in all different shapes and sizes. Well, most of them are small, hundreds of nanometers, but they come in all different shapes, and some of them are bigger than others. The influenza virus, like the SARS coronavirus or the COVID-19 virus, is an enveloped virus. Has a structure like this one. And then there are all different kinds of viruses, including really nasty ones like the Ebola virus over here that form these long, stringy, filamentous forms. And then there's even these really fancy ones that look like Sputnik or lunar lander module, the bacteriophages. And we're going to talk about these for a second. Okay, so these are viruses that infect bacteria. Okay, so they, oops. Let's see, I got to slide out of order here. So these viruses infect bacteria, and they do so by landing on the surface of the bacteria and injecting some genetic material, some DNA into the bacteria, or RNA into the bacteria. And in doing so, that's one of the key steps in that virus being able to then take over the cell and replicate. Many bacteriophages are lytic, meaning they'll take over the bacteria, replicate themselves, and then eventually the bacteria bursts and releases a whole bunch of new bacteriophages. But one key piece, one key piece of information is that they inject a piece of DNA or RNA, genetic information. So right now, you've probably heard a lot about COVID testing and how that involves sticking a swab up your nose. So if you had the coronavirus infect cells in your nasal cavity, that little swab can be used to get out some of those viruses, which are then put in a chemical that breaks them down, freeing up some of their DNA and RNA. And that can then be placed into a PCR machine, or polymerase chain reaction machine, which can amplify that DNA. And if there's viral, like COVID-19 viral DNA or RNA in the sample, it will be amplified. And if it amplifies well, we know that you had the virus in your nose and that you would then have a positive test. So that's one example. Now, that's one of the ways that we as humans help defend ourselves against the virus. Bacteria, they don't have this kind of technology, but they have evolved their own approach, their own technology. And that approach was called CRISPR, which is a funny acronym, clustered interspersed repeats. These, and I'm going to just describe briefly how the CRISPR system works in bacteria. And what it is it's basically a defense mechanism against the bacteriophage viruses. Okay, So these bacteriophages have to inject a little bit of DNA, phage DNA. And the first stage of the CRISPR process involves recognizing that foreign DNA, snipping a piece of it out, and then copying that piece into the bacteria's genome, into its chromosome. Kind of like those retroviruses I mentioned. They copy their own DNA into ours. But in the CRISPR system in bacteria, they write a copy of a portion of the viral genome into their own bacterial genome through this segment here where you have these clustered, regularly interspersed, repeating regions. So you have spacers and then repeating fragments of the bacteria, of different phage or bacteriophage portions of their genome written into the bacterial genome. What that allows is when the bacteria gets infected, say a second time, or a progeny bacteria gets infected, this CRISPR region is being transcribed. It's making little pieces of RNA, DNA-like RNA molecules. Those RNA molecules get docked to this molecule called Cas9. And these RNA molecules are complementary to the phage DNA from which they came, so they will stick. And when they stick, that brings the Cas9 protein to the phage RNA, and it cuts it. Cas9 is a molecular scissors, guided by a copy of the virus's own genome that the bacteria has been hanging on to. So CRISPR, basically gives the bacteria a way to make themselves immune. It's like a primordial immune system for bacteria to infection by bacteriophage viruses. So I think, the next section of the talk, I'm going to talk about how we've taken as scientists, these two scientists here, Jennifer Doudna and Feng Zhang, have taken the CRISPR tool and re-engineered it. It made it into a very powerful tool. But I think before we do that, we've got a game to play which we can protect the bacteria from infection by bacteriophage.
Yes, so I put the link out in the chat a little bit ago. So there is a game. I'll put the link back up again for those of you who are just joining us. But basically, this game can be downloaded onto any iOS operating devices. There isn't an Android version yet, unfortunately, but you can play it from a browser. And so I'm going to share my screen and get the game going. Let's see. All right, can you guys see the game?
Yes.
Okay, so now I need to make sure I'm in the right spot to play it. Here we go. I'm going to turn my volume down. Okay, so the game basically and I encourage other people to load the game and play, especially Mesa and Race if you want to play, or you can be just watching, but then be asking questions. Then people who are watching via Facebook, YouTube, or Twitch, you can be playing the game too. You can be commenting, asking questions. And we're just going to Adam's going to talk about how this kind of relates to what he just shared and how the game is modeling what he just talked about with the bacteria. What I'd like you to do, those of you who are watching, is what do you notice in the game when you're playing and thinking about what was just shared about CRISPR? And what are you wondering, right? Because we haven't really kind of gotten to like the spoiler of how this is being used to help fight COVID, for example. And feel free to add those comments and questions, those notices and wonders in the chat. And then Mesa or Race, I can't see the chat right now because I'm playing the game. So you guys need to be typing in the chat and responding to people as we do this. So here we go. So there's an army of ruthless phage invaders. These villainous viruses want to infiltrate bacterium and hijack its resources to build more copies of their lifeless, lethal soldiers. As neighboring towns fall to phage forces, the survival of bacterium is in your hands. Cellular streets towards the viral DNA and cut it apart. I think I just cut off part of this. Hang on a minute. Use the mouse or trackpad to move through the cellular streets towards the viral DNA and cut it apart before these genetic instructions are used to construct a new piece of a phage soldier. If all three parts of the phage are built, they will assemble into an unstoppable phage that will burst out of the bacterium, causing damage. If enough phages replicate and break through the walls of the bacterium, you lose. All right, here we go. This is
okay.
This is really hard to do without my mouse. I left it at home.
Yeah, there you go. You did it.
All right. Phew. I was able to fend off that red phage pretty quickly. And there's a different kind of phage approaching the cell wall. I will have to make sure I'm carrying the right guide RNA each time.
So the guide RNA is what targets Cas9. So Cas9 is the molecule that you're controlling, I guess, inside this cell. So it looks like the blue guide RNA has to match the blue phage genome. And as long as they do, that Cas9 can match up with it and cut it, cleave it, destroying it. Hey, you did it again. You're pretty good at this, Bree.
I practiced last night.
I did too.
No, it's slow.
Oh, that phage is growing. Oh, it's got legs. I found that you have to kind of like click back on the Cas9 again. Well, you still did it. The phages were replicating, but you got it.
All right, this says to watch out. So this is, notice like what Adam was talking about before when he shared how they insert the pieces of the DNA or RNA from the phage into their own plasmid or genome. This is kind of what that's modeling in here. So now I'm gonna start again. So there it is floating around in the cell. Makes it a little harder for me to get to the...
Yeah, you start to collide with it, right? I love how these phages, they just sort of like little rocket ships and they just take off and fly out of the cell. There you go.
All right, so yeah, the bacterium is storing all this RNA and but it's starting to get in the way, right? It's building, it's taking up space. So that's kind of what we're seeing now. I'm gonna try this with my arrow keys. I feel like it's not working.
Yeah.
This is like back when I was a teenager myself trying to play like Grand Prix race car games on a PC computer with just the arrow keys or Tetris.
There we go. Yep. So you can see that. So that thing that's slowly the train circling around in the cell there, right? That would be the CRISPR itself, the clustered regularly interspersed repeats.
I died.
Ah, fatality. Yep. So I think, I managed to get to the place where pretty soon the phages start shooting in little anti-CRISPR, anti-Cas9 machinery that starts to gum you up. Maybe like the next level. Which there is also known that some phages, some viruses make molecules that will inhibit this system. The Cas9 system, and that's true. So that same thing is seen in viruses that infect humans. So viruses like the coronavirus play all kinds of tricks on our cells to prevent them from blocking the early interferon responses and the immune response, from destroying them.
All right, Adam, we actually have our first question, which I think is a good segue to you explaining what you're doing. The question is, how is CRISPR being used to study other infectious diseases? And is this being used as a therapy for any other diseases?
Yeah, great questions. I'll take the last part first. As far as therapy, there are some initial work being done to try to use CRISPR to reverse mutations or change mutations, such as sickle cell anemia, which is one little change in the human genome causes that disease, and a person could potentially use CRISPR to reverse that. And then we'll talk about infectious diseases here. I'll give some examples, or I'll give the example of some of the work that we're doing and how we're using CRISPR right now. So much of what we're able to do was enabled by these two individuals. Jennifer, those are great. Yeah. Okay, Jennifer Doudna, and she spent a long time trying to better understand the CRISPR system in bacteria, and then realized that there was a technology that could be created. So it's fairly difficult in molecular biology to take and make changes to a genome that's already there. Big, complicated. It's like, imagine those big, complicated blueprints, and you want to make a little change, like you want a door in a different spot, or you want to put, tweak to a house plan. That's actually was previously a pretty tricky thing to do. But what Jennifer Doudna and Feng Zhang, professor at MIT, recognized was that the bacterial CRISPR enzyme, in particular the Cas9 enzyme, could, if it was modified, we could write little instructions into the guide RNA, basically use the guide RNA, which would write like a little instruction that would tell the Cas9 enzyme where to go and where to cut. And so they really brought this from understanding how the bacterial immune system, if you will, works into a tool and a technology that we can use today. So what they had to do in order to do that is they had to understand the molecule, how the molecule Cas9 worked. And so they had to go to a bacterium that had a fairly simple CRISPR system in it, take the Cas9 molecule from that system and really understand its inner workings. And what they realized, one of the key innovations is they realized that they could take this guide RNA and they could combine it with, it had two parts. They were able to combine those two parts and create a single guide RNA. And that single guide RNA will then stick to the target DNA. It'll the word we use is hybridize. It's like two halves, the orange, the kind of yellowish orange, and the red half have to come together. And if it does that, that brings this protein here, this Cas9 protein, to that site. And that is, the Cas9 protein can then cleave or cut the target DNA. Now, what is that? Okay, great. So we can cut it. What does that do for us? Well, it does a lot. So there's a simpler cartoon of this process. The Cas9 enzyme is brought into place by its guide RNA. It has these two little scissor-type pieces which can cleave the target DNA. And in doing so, it creates a break, double-strand break. Now, two things can happen once that break is created. One is this process here called NHEJ, non-homologous end joining. And in this process, the cell will stick the pieces of DNA back together, but it's really error-prone. And so when it sticks them back together, you'll often get insertions or deletions. And if you do that in the right place in the genome, you can use this to shut genes off. Okay? It's a really effective way to turn a gene off. And that's mostly how I'm going to talk about it and how we use this thing to shut a gene off. The other thing that can happen is you can get other types of repair. So there's other tricks that the cell can use and that we can play as molecular biologists to get cells to repair the gene differently. And you can see this one here. It's called homologous recombination. Here we provide a donor piece of DNA that maybe has a change in it that we would like to have. Okay, like the question about is CRISPR being used to treat diseases? Well, one way is to use an approach like this, where the Cas9 enzyme cuts and makes this break. And then delivery of a donor DNA can be recombined at that site, creating an exact transfer of that sequence. So in other words, we have then edited, we've rewritten the instructions that were in that gene, changing. So I gave the example of sickle cell anemia. It has a tiny little change in it that causes the disease. If we use an approach like this, we can overwrite that change or correct that mutation, and in doing so, repair that gene and repair the function. Okay. So now I want to talk about how we're using the CRISPR system the CRISPR technology to understand COVID-19 and the virus that causes COVID-19. It's gonna focus on this first part here, this error-prone NHEJ process, which is really good at shutting genes off. Okay, so you can pick out genes that you want to study, and you could take your CRISPR protein and make a guide RNA. It turns out these are very easy to make. There are companies that make them. They have machines that do the chemistry and make them very quickly. Usually we can get them in maybe 2 days time. We can have that piece of RNA that's needed to send Cas9 somewhere in a genome. So what you can do is you can sit down at your computer, design some guide RNAs, company ships them to you. You can insert them into a cell along with Cas9, and you can shut off that particular gene. Those are called targeted knockouts. So if you have a hypothesis about, I think this gene affects some biological process, you can use CRISPR to very quickly test that hypothesis by going into that cell and shutting that gene off. Now you can imagine scaling that up a bit, right? Like, okay, let's look at maybe a few genes, 10 genes or 100 genes, right? We call that a rational screen, and I'll explain what a screen is in a second. But it turns out we can pretty quickly make a number of these guide RNAs to target a bunch of genes or a handful of genes. And so we could do a bunch of individual experiments to test those ideas. And then there's the last idea, which is called a whole genome screen. And that's what I'll talk about today. So the strategy here is, okay, we want to understand which genes out of all of the genes in the human genome affect a process. In this case, affect the process of the coronavirus infection. Okay? So in that case, we need to go in and inactivate every gene one at a time. But there's a problem. There's like 20,000 of them. Okay, so there's quite a few we have to deal with. But some clever folks have figured out how to do this, and our lab has also figured out how to adapt these methods and become very efficient at it. So what we do is we can make what's called a library of guide RNAs. So we have a guide RNA. Actually, in general, we have something like 4 to 8 to maybe even 12 different guide RNAs that target each genome or each gene in the human genome. So it's like a big collection of these things, like 100,000 of them or 200,000 of them. And they're all mixed up. Okay, it's just a pool of them, but it's like a library, but nobody organized the things. Okay. And so we have them, in like a test tube, right? And they're just all mixed up in there. So what we can do then is we can play some tricks, and this is like, I'm just going to talk about how we carry this out. So the CRISPR whole genome screen, we have the Cas9 molecule, and we express the Cas9 protein in a cell. We have to basically engineer these cells by adding in the Cas9 gene. So we do this in cells in a dish, right? Cells that we've cultured, either immune cells or other cells, just working with cells in a dish. And we can engineer them to express Cas9. Then what we do is we take advantage of a different kind of virus, a retrovirus or lentivirus, which is really good at inserting little pieces of DNA. And we use these lentiviruses to deliver our guide RNAs, okay? And they actually insert a gene, a little tiny gene that then leads to expression or production. So we use the virus to produce the guide RNAs that will then target the Cas9 enzyme to the gene specific for that guide RNA. Now, in this lentivirus, we can use these lentiviruses to deliver the whole library. So, in other words, we'd have like 100,000 different lentiviruses with many, many copies of each one of them in a test tube, in the tube, right? And we put them in a very small quantity, such that each cell only gets transduced, is the word we use, with one lentivirus. So they each get one different guide RNA expressed in them along with Cas9. So in other words, each cell has a different gene disrupted in it, or it's random, but overall, on average, they have distinct gene disruptions in them. Okay, so what we've done then is created a pool of cells that have either lost functions or gained functions depending on which genes have been shut off. We take these cells and we challenge them. Right now, we basically challenge them with the coronavirus, but we're not actually using the coronavirus in our lab. We're using a modified fake virus, if you will, that has the same surface properties, same surface molecules as the coronavirus. And it's engineered to deliver a gene that codes for a red protein, a red fluorescent protein. So if the cell gets infected with this virus, it will produce the red protein and fluoresce red. Okay? And cells that are have genes knocked out that are allow it to be infected more easily will be more red, or more frequently they will be red. And cells that are have genes that are required for the infection shut off will have a hard time being infected. They'll be protected against the virus. So what we then do is we sort these cells. So we have a machine called a fluorescence automated cell sorter. And we basically take these cells, usually about 30 million of them, and we flow them in a tiny little stream. So using kind of a microfluidics type approach. And that stream creates little tiny droplets. And those droplets are then sorted between plates carrying an electric field, and the droplets get an electric charge, and this machine can rapidly sort the cells that did not get infected or were protected away from the cells that got infected or highly infected, which now have that red fluorescent signal associated with them. So we get two tubes. We get one tube that contains the low infection cells, or poorly infected cells, and we get another tube that has the highly infected cells in it. And of course, we have the original population of cells as well. We then turn to the same kind of technology that was used to sequence the human genome, a little bit smaller scale. This technology is called next generation sequencing. And if you look at the machine, it looks just like a box. But this box does an amazing thing. What it does is it will, for every one of those cells that we sorted, we get a copy of its genome in that tube. And we use some chemicals to disrupt the cell and get the genome out. And we amplify it using PCR, like that detection method for COVID that I mentioned a little bit ago. But we amplify the little pieces that were inserted by the other virus, the lentivirus. And we use this machine to then read off the guide RNA sequences, those which act like barcodes. So if we read the guide RNA sequence, we can match it back to our library, and we know which gene it targeted. Okay. Now we have to take a lot of data points. So usually in an experiment, we will get something like 200 million reads. So 200 million pieces of information about which guide RNA was in each sample. Maybe 30 to 50 million reads per test tube sample out of that. A lot of information. And then we have to use computers to do statistics on those barcodes to map them back to the genes in the genome and ask which genes were enriched and which were depleted from the high infection and low infection groups. Okay. And in doing so, we can then start to associate gene functions, with the susceptibility of the cell to infection or protection of the cell to infection. So right now, thanks to a grant from the National Science Foundation, my lab is, we use this method to do lots of things with the immune system, but we're using this system to better understand how the COVID-19 coronavirus infects cells and what are some of the genes. So we'll talk for just a second about some of the things we hope to learn from this. We hope to learn a little bit more about some of the machinery that's involved in allowing the virus to attach to the cell surface. We know some of the receptors, but there are likely more. There are also other pieces of machinery that process the viral envelope protein. They cleave it. There's a lot of cutting and snipping in biology. They cleave it, and that cleavage activates it and allows the virus to then fuse its membrane with the host cell membrane and deliver its contents into the inside of the cell. There are also genes that are involved in bringing the virus into the cell, and also genes that are gene products, protein machinery that are involved in helping the virus be released from those endomembranes, and ultimately would allow, in our case, the reporter, the red fluorescent protein, to be expressed. So those are some of the key things we're hoping to learn about in the system we've developed. We do think that eventually we could transfer this technology into an environment where we can use a biosafety level 3 lab and run the same kind of experiments with the live coronavirus with all kinds of protective gear on in that space to prevent us from being infected. So with that, I'm happy to.
So does that mean, Adam, does that mean then that basically you could then use this in a therapeutic way, like with sickle cell, where if you knew that you could turn off a gene that would prevent it from invading a cell, like could it be then used therapeutically to like reduce that viral load or the risk of getting more, like, not necessarily, I suppose it might be used for prevention, but it's really more of like you have it and now you can use this as a therapy to make sure you're not getting as sick. So it's like basically, it ends up kind of being like an antiviral type of therapeutic. Is that what how it would be?
So there are some people that are working on using CRISPR as an antiviral type therapeutic. For the coronavirus, it's kind of hard to see how that would really work, especially if you were talking about editing the host cell itself. Those are very difficult treatments to get to work well. So examples like cystic fibrosis, where people have a mutation in their lungs, in certain gene in their lungs that causes that disease. People look at CRISPR as a potential way to treat that. For an acute viral infection, though, I think it would be difficult to come up with a way to do it efficiently and cost effectively at this point. But who knows? I mean, that's why we need new scientists working on these problems and coming up with new strategies, because it might be more feasible than you might think.
All right, Mesa or Race, do you guys have some questions you want to ask Adam? And I'm waiting for questions to come in on the chat. I am going to, so we do have a Kahoot that we've created to see if people, what you learned about CRISPR. So I'm going to put the link to the Kahoot in the chat. Oh, wait, here we go. Let me resend that. All right, so if you want to join the Kahoot and play that, I'm going to share my screen. So you can join the Kahoot. We'll see if people join. All right, we're going to get started. What factors influence someone's chance of getting COVID-19? All right, here comes the next question. Nice job, Mesa. Okay, what does CRISPR stand for? Woohoo, I'm in the lead. All right, here we go. Next question. How can CRISPR be used during the COVID-19 pandemic? All right. What is a benefit of CRISPR? All right. Gotta turn that off in the background. Okay, well, thank you to those of you who are playing the game. And that was actually a really great way to review what we've been learning about tonight. And so it is almost 7 o'clock. If anybody has any questions and they want to add them in the chat on YouTube or Twitch or Facebook, please type those questions and or comments, and we will see if we can answer them for you. I think while we're waiting, I guess I'll ask you a question, Adam. Can you, as you were talking about your career and kind of how you landed where you are, can you think of a particular moment where you faced, what's been the most challenging thing for you in terms of like, and especially, I think, actually, I'm going to reframe that question. So thinking about the current situation we're in, especially when things kind of shut down in the spring, what has been the most challenging thing for you as a scientist in trying to do the work that you do in the midst of this pandemic? How has your work changed?
Yeah, I mean, I think I can almost answer maybe both of those, about the historical challenges too. As far as the pandemic goes, being someone who's trained in infectious disease and immunology, I think a lot of us who've studied viruses and been in this field for a long time kind of knew this was coming, right? And we've always been watching for it and waiting, and to see it hit. And I think we also got used to the idea that the systems we had put in place, the Center for Disease Control and the World Health Organization, really had done a fantastic job of locking down other kinds of outbreaks. And so to see those systems just crumble or not be able to hold the bulwark against the coronavirus that was a real challenge, just to even watch that happen. And then second, knowing that we know a lot about these types of respiratory viruses. Coronaviruses haven't been that heavily researched, and just kind of knowing that this is going to be a real challenge, and it's going to take a lot of people coming from different areas of virology and immunology and medicine to come up with real solutions. So I think that was one of the challenges. And then another one for me was trying to figure out what's the best thing that we should do. So I'm involved in local advisory groups and so forth, where we look at data and try and understand the trends in our area. So very regionally, trying to understand how the response is being managed and what are some of the policies and ways to think about policy that can help with the pandemic, as well as being involved directly in the science. What's the best scientific things that we can do? I've mentioned that I work a lot on the immune system and therapeutics. So therapeutic antibodies are one of the things that's being used to try and treat COVID patients. For example, the president just received a pair of antibodies from Regeneron as part of his treatment therapy. That's the type of work that we do. So trying to think about that, or is it using our CRISPR, the screening approaches we've developed in CRISPR. So trying to figure out that landscape and what there's, you feel, to me, I felt like, oh my gosh, there's like a million things I should be doing. And I spent a lot of days early in the pandemic just reading papers, all day long, trying to map out strategies for different ways to help. And I think a lot of scientists with the type of training that I have, we're in that same boat. Like, it's kind of, oh my gosh, there's like 30 things I could try to do, but which one is the right one to do? So that was a real challenge. I think I'm pretty happy with what we decided on and set out to do. And then I think you kind of asked another question at the beginning of it. I think you were asking about the career trajectory part and the biggest challenges there. And I think that's a good question. So even if you didn't mean to ask it, I'm going to answer it. So everybody thinks differently, and we all face challenges. For me, I was like as a kid, I mean, I was ambidextrous, dyslexic, and, learning disability diagnosed. I'd struggled a lot in school because I just didn't learn like everybody else. And that was a struggle, thinking about high school and even into college, that I grappled with. And I think part of, being a successful scientist isn't, it takes hard work and not giving up, when people are might be sticking some kind of label on you. So if you're excited about it, your creativity's there, I think it's really important to just go after the things that make you happy. You'll find a way. I mean, for me, that dyslexia and so forth actually became, I think, one of my strengths later on. So some things that people think are weaknesses turn out to be, can really be a strength if you figure out how to nurture it. So I just wanted to answer that one because I think for me that was, if I had given up and felt that I couldn't do it, I certainly wouldn't be doing what I'm doing now.
All right. Mesa or Race, is there anything that you noticed tonight with the presentation? Something you're wondering about? Any last minute questions? What did you learn?
What did you think about the people that, I put a couple of pictures up of some of the folks that, these two folks, they contributed a lot to taking, understanding and turning it into a tool.
So I noticed on the video game page that one of the scientists, she actually has a YouTube video of her playing the game. So was she involved in actually creating the game, do you know? Or did she just get asked to like give her 2 cents about it?
I think they asked her to play it. That was how I interpreted it. Yeah.
All right. Well, so speaking about games and other really cool kind of apps that you can explore to learn more about CRISPR, I'm going to share a couple resources and then we'll kind of close this out tonight. So I'm going to share my screen again. So there's actually this really cool app you can get on your phone. Just put in a search for CRISPR 3D. And it's actually developed by the same people who created the game that we played earlier. And actually on this website, if you go up to education and then go to games and activities, you'll see the Phage Invader game. And then there's that 3D app. And what's really cool about it is it basically is kind of, and if you have any virtual reality goggles, they actually have an app for that too. But basically the app has, you can see there's a bunch of, they have an image and a model of all of these different Cas molecules, these Cas proteins, and Adam talks specifically about Cas9. And so these all kind of play a role in that kind of molecular chopping of or cutting of the bacteria or the virus, the bacteriophages RNA, but they're not all equally useful in terms of how we use them in biotechnology to solve human problems. And so Cas9 is kind of that's like the best one, right, Adam?
That's the one that's really like the technology, enabler. Yep. Yep.
And what was the other one? That's like the next best?
Well, so there's also another molecule called Cas12, which is not as well developed as Cas9 as a tool or technology, but it's becoming increasingly, developed, I guess.
All right. So this app it looks like it has 13 of the Cas molecules in the models, and it's really cool because you can see all the folding and everything that happens in terms of the shape of the molecule. And then you can hold your phone up against a surface like the table, countertop, or whatever, and then that molecule just kind of pops up into your world. There's a card game too, by the way, that I feel like I'm going to have to go order. But this is really fun. These are examples of people posting on Twitter or Instagram with their Cas molecule, and then their comments like, Cas9, is that you? And anyway, it's just kind of fun. So you can access that app and you can kind of play around with the molecules and learn about them. And we encourage you to share your nerdy posts of you incorporating protein molecules into your daily life. And then there is another game called CRISPR Defense, or CRISPR Defense or Defender. And that one we were going to use tonight, but it did not have, there's no desktop option for that. So we couldn't stream and play it simultaneously. But I encourage people to look that one up because it actually gives you a really good tutorial on what Adam talked about tonight in terms of how the bacterium identify that piece of RNA and then make copies of it. And then you can play a game like that other one where you're trying to protect the cell from the invasion. Mm-hmm. So let's see, we have another question, and it is, could you use this type of information in a dental setting as well? I'm a future dentist / researcher. Mm-hmm.
So I think, the sky is really the limit, and the, creativity is so important. So I mentioned, so one of the CRISPR inventors, CRISPR technology inventors, Feng Zhang, and actually Jennifer Doudna, they both have companies that work on technologies based on CRISPR. And Feng Zhang's company came up with a really clever way to use CRISPR to detect the coronavirus, right? So they have a rapid diagnostic that they've developed, and it looks like it works reasonably well, that uses the CRISPR enzyme along with some special guide RNAs that allow things to change color when the coronavirus is there. So that would be an example. So you could imagine using it, as a diagnostic or some kind of tool in dentistry to measure something, maybe like for some of the bacteria that cause cavities, strep mutans, these the bacteria that are involved in causing cavities and things like this. So certainly, I think there are the number of applications for this molecule are really impressive and really far-reaching. Not to mention just studying the immunology and the immune system of the gums and the teeth is a fascinating subject all by itself, in which CRISPR could be used to study that.
Any other final questions on the chat? I'm just checking really quick, and I don't see any. So there is a link that was just put out to the chat for a Google form for participants to take a survey to help us learn what you learned and to help us do better next time as we learn how to do these virtual cafes. And then actually there is, here's a new comment. It says, if we were interested in looking into more research that is being done on how we can use CRISPR technology to fight new diseases like COVID, are there any papers, research, or readings that you would recommend?
I don't have a short list ready to hand out. I can make one though. Okay. One of the, so if you wanted to post it, we sure could do that. Right. What I would recommend though is, so kind of the two premier journals, Science and Nature, along with Scientific American, have pretty consistently putting out papers, not research science papers, but summary review articles that talk about how CRISPR can be applied and how it can be used in different contexts. And so I would recommend looking into those three publications. So Science, Nature, and Scientific American. There's a number of good articles in those three journals.
All right, so we'll make sure, Adam, if you have some other resources you want to send me in an email, I will put those together and share those via our Instagram account and the Discovery Center's Facebook account. So for those of you who are watching who are new to this program, you can follow us on Instagram. The Teen Science Cafe program specifically for the South Dakota Discovery Center is called STEM Savvy. And they have an account you can follow on Instagram if you look up stem.savvy. We're also on Twitch at STEM_savvy, and there is also a STEM Savvy Facebook page. And then, of course, the South Dakota Discovery Center has a page where we actively are posting information. And then I'm going to share my email in the chat. So if anybody has any further questions, they can email me and I can follow up on that. If you want to know more about when this leadership team meets, so you can get involved in planning our next one, or if you have questions for Adam, then you can email me and I can get that information out to the people that it needs to go to. So thank you for your time tonight, and thank you to our viewers who've been watching. And please, again, finish that survey if you can, because it helps us do what we do better each time that we do it. And actually also, too, this isn't in the survey, but if you are watching from Twitch or YouTube or Facebook, I'm really interested in knowing about what that experience was like. Were there any glitches? What would make it more interactive? So please feel free to comment and or to email me with that information so that we can do this better next time. Thank you.
Thank you so much for having me.